Good evening, everyone. My name is Mamta Gupta, and I'm currently doing my Masters of Artificial Intelligence from Queen's University. I'm an aspiring data scientist in Toronto area. I welcome you all to today's talk on AI-aided design of novel inhibitors against SARS-CoV-2 by Dr. Zhu. Dr. Zhu is Shoemaker Endowed Professor in Department of Electrical Engineering and Computer Science. He is the Director of Information Technology Program with appointments in the Christopher S. Bond Life Sciences Center and Informatics Institute at University of Missouri, Columbia. He obtained his PhD from University of Illinois in 1995. He was a staff scientist at Oak Ridge National Laboratory before joining the University of Missouri. His research is in computational biology and bioinformatics, including machine learning application in bioinformatics, protein structure prediction, post-translational modification prediction, mobile app development for healthcare, and many more. He has published more than 360 papers. He was elected to the rank of American Association for the Advancement of Science Fellow in 2015 and American Institute for Medical Biological Engineering Fellow in 2020. This being said, I will pass on to Dr. Zhu. Over to you.
Yes, thank you for the introduction. I also like to thank the organizer for inviting me here. Toronto machine learning community is very well known. It's an honor for me to give a talk to the community. Today I will talk about our recent work by using AI for designing novel drugs targeting COVID-19. So we actually published work in bioRxiv recently. Here show the link. This work is mainly done by Bowen Tang and my lab. This is also in collaboration with Professor Zheng Wu at Xiamen University in China. So for COVID-19, I know in the introduction, I just want to point out the underlying virus called SARS-CoV-2. This virus actually codes in proteins. So if you look at the virus itself, it actually has this structure shown here. Some S protein, surface protein, inside RNA coding of protein. Any of these proteins from target, that is the small molecule, by protein, tightly with the deactivate protein function, cure the virus. So you have tried the different targets with them both S protein on the protein on the virus surface. Not against this. But unfortunately, this protein is not a drug or vaccine because it has a sugar molecule. Other molecules, people look into this so-called 3C-like proteinase a lot because this protein, even though it's not expressed on the virus itself, but it's very important in the viral process. Basically, the protein cut long peptides into smaller proteins, and in HIV, this is a key protein most successful HIV drugs target. So here for this virus, people also think this is the main target. This protein is also called M protease. So we use a M-Pro to represent this protein. So the simple thing that we could repurpose some existing drugs for targeting this protein. People have tried the HIV drugs, even though these drugs have been very successful against HIV viruses, but it's actually shown not effective to target COVID-2 viruses because the structure of M protein in HIV and the M protein in COVID-2, they are dramatically different as shown here. Other inhibitors, people also tried. So far, there's some promising candidate, but there's still no proved drug against COVID-19. That's why it is it is effective probably to design some novel chemical structures. So one source of information we could use is SARS. So SARS happened about 17 years ago, and it has a very similar sequence like COVID-2. In particular, this M protein, their sequence and structure are very similar. Here shows the structures of SARS protein and COVID-2 protein. They can superimpose very well. And over the past 17 years, many ligands have been designed to target M protein of SARS. We actually collected about 300 active inhibitors. So these inhibitors cannot really be regarded as a drug because they have not been approved by FDA, but they have shown some activities. That means they have some binding demonstrated by the so-called IC50 numbers. So we can use these inhibitors as a base to design novel drug to target SARS or SARS-CoV-2. The basic idea that for the 300 molecules, we can partition them into smaller chemical groups, and then we can recombine them to generate the novel chemicals, and then to test whether they are suitable candidates. If you look at for example, one of the promising drug designed so far by others, it can be actually combined by these chemicals. So this is a new one, it's called N3. So the way to do it is to use existing chemical knowledge. They are represented by SMILES, SMARTS, and SMIRCs for chemical, this formula for chemical reactions. The idea is the following. So let's say you have these existing compounds, and then we can break them into meaningful units. So each unit has some basic standalone chemical features. For example, you cannot break a binding into half because half binding is not meaningful. So you can have these groups and then recombine these groups to form additional chemicals. Of course, when you form additional chemicals, you also have to follow these chemical reactions that's actually encoded by these SMIRCs. So this recombination, in our case, we only consider recombination of 2 units. And then we actually define these reactions into 46 types, and they are grouped into 16 categories. They are encoded by such SMIRC representations. So this representation you can view as its underlying chemical knowledge and chemical reaction possibilities or rules. So here shows this example how they are encoded. So at the bottom, they are basically individual atoms like a carbon, nitrogen, etc. But the upper level, you can have even more property associated to it. For example, this could be aliphatic nitrogen. That means that not only it is a nitrogen atom, but it also has certain chemical bond information. And then you can go to the next level. That means that you can have these chemical groups, and then they can combine into different units. So here shows this example. You can actually have this group of chemical and this group of chemical. This is just a general formula. This one star here is a placeholder. It could be carboxy group. It could be a chloride group. So this basically represents one group of a molecule. And here it could be another group of molecule. And then they can combine to have different scenarios to form this final product. So by encoding such knowledge into the SMIRCs encoding, we could use a machine learning method to systematically explore all these possibilities. And the way we do it is through reinforcement learning. That is the same method for this Atari games. So in the Atari game, you have this machine learning network to assess so-called value function. That is whether you are in a winning position or what action would likely to be a winning position. And then this is the agent to train a model so that your action, or in the machine learning term, it's called policy, the more likely to be successful. We do the same here, except that we do it for drug discovery. We train a reinforcement learning agent to recombine these chemical groups and to form new compounds, and then to see whether these new compounds are good potential drug targets. Of course, for this reinforcement learning, the first thing we need to define is so-called reward function. That is the final molecule we designed, whether they are suitable or effective molecules against those proteins. So we actually collected all those 300 molecules, and then we dox them. We see potential patches that are important. And we use this tool called RDKit to see the important chemical groups. There are basically two types of chemical groups important. One is so-called hydrogen bonds. That is, you have a donor receptor between the small molecule and the protein, so that they can form stable chemical hydrogen bonds. The other one is hydrophobic patches. That means if your molecule have a hydrophobic group covering a hydrophobic patch on the protein, then it's more likely to be stable for this binding. So these positions can be represented by dots here. That means our designed molecule should cover as many of these dots as possible and should satisfy those conditions as much as possible. The condition, I mean, either hydrogen bonding condition or hydrophobic patch conditions. And then the idea that we have this initial molecules, we break them into small fragments. In our case, each fragment is less than 200 Dalton, so it's not too big. And then we can recombine these fragments through this library we generate by this reinforcement learning. And then we generate the chemical molecules as candidates. Then we keep searching to find good molecules. Of course, other than reward function, I just talked about for hydrogen bonds, for hydrophobic patches, we also consider general chemical properties like a QED or CSF to make sure those molecules, they make sense, they are relatively easy to synthesize. One thing we actually modified from the Atari network is to apply a more cutting-edge reinforcement learning framework. It's called a dueling double deep Q learning network. So this dueling network in recent years shows really good performance. The idea that you have two channels. One channel is to train the value function. The other channel is train the advantage stream. And then you can optimize these parameters so that it makes the agent stronger to find optimal solutions. In parallel, we have another targeted dueling deep Q network. This is almost like a mirror of this, except the parameters here are updated slowly. Here we update these parameters every steps during this reinforcement learning search. Here we only update every 5 steps. By using a fast update network and a slow update network, we can search more effectively, and also the search tends to be more robust. And then here shows this fragment-based search. So there are actually some other reinforcement learning methods to grow molecules one atom at a time. But we are really the first framework to grow molecule one fragment at a time. By doing that, we can grow more effectively to have faster search, but more importantly, our chemical generated from this approach makes more sense chemically, so that it's more likely to be effective. So the way we do it through this reinforcement and search, that is, we have an initial structure, and then we do this exploratory actions to look into many possible combinations of fragments. And then we identify a good one, and then we keep searching it, and then we update the good conformation with better and better so-called value functions. And then each time we start with those better conformations to expand the search tree. So we do this iteratively. That would allow us to really find highly effective target. So target we find not only effective in terms of general binding. Another idea we apply here is that we use a so-called bonded interaction. So this molecule not only bind to this active pocket, but there actually is a cysteine residue 145 on the M protein that potentially can form chemical bond to this ligand. So we use this chemical reaction shown here to make sure that our chemical group can align to that cysteine 145 properly, so that there is a good chance to form strong chemical bond. And this reaction doesn't require enzyme, so it's a non-enzymatic reaction. And by having a bond interaction, it will attach to the M protein very strongly and more likely to inhibit the function of M protein. So in order to do that, we actually make the docking structure, and then we simulate the binding. And then after this simulated bond interaction, we optimize to see whether the conformation remain the same. If it's similar or very almost the same, that means it's readily available to form this chemical reaction. Of course, we sometimes need to do some adjustment in order to achieve that. One thing, for example, we do here that in order to have a better placement or orientation, we add one carbon on top of this. So we could do this small adjustment to make sure it binds better, it has a more chance to form this bond interaction. We also make sure that at the quantum mechanical level, it can form a strong bond interaction. So we perform this QM-MM umbrella sampling on this molecular dynamic trajectory to make sure that they have a very good chance to form a bond interaction. And finally, we can get some good candidates. And then we test these candidates through molecular dynamic simulation by checking their binding free energies. So here we compare among 4 cases. These 2 are HIV drugs. As we already shown, they are not effective. So they are sort of the false positives as control. This N3, it is a good candidate. So it's a positive. And here's the molecule that we designed through this machine learning methods. So we run this molecular dynamic simulation to calculate free energy to see how stable the conformation is. We found that these 2 HIV candidates, their binding energy is not very strong. This N3 is a new drug, indeed has a much stronger binding energy. But ours has even stronger binding energy than N3. And also, if you look at the RMSD, so-called the conformation fluctuation, ours changes in a relatively narrow range, even without a bound interaction. That means our binding is more robust. So we actually performed some experimental validation. This is done at our collaborator, Professor Zhong Wu's lab at Xiamen University in China. So they actually synthesized our predicted candidate. And this basically is a synthetic pathway, basically all this reaction. And here lists all this enzyme that leads to this final compound. We found that we have to adjust a little bit. It's not quite the same as initial design, but it's just a very minor modification. But we believe still it is close enough to have a very strong binding. So in this work, we have not really optimized the best synthesis. So that could be our future work. So in summary, we developed this deep Q network method using this fragment-based approach for drug design. So the method is novel and also is effective. And we use it's a dueling double DQN, which is a cutting edge. We believe it's more effective than other machine learning framework. And also, we encode chemical knowledge in machine learning. And also, we grow the fragment one at a time instead of one atom at a time. This has not been done by other methods. We also use previous drug design data, that is 300 compounds from SARS drug design. So this gives us a better basis for drug design. We also extended this method to other design. For example, we designed some peptides in addition to small molecules to against various proteins. And our collaborators are testing those peptides as well. The idea is actually very similar, except the peptide is easier to synthesis. Of course, the flip side is that the peptide has a less choice to search optimal combination. So the binding is not as strong as a small molecule. And then we designed all these small inhibitors for COVID-19. We believe these inhibitors are more potent than any other drugs we know. That means that they have a very strong binding demonstrated by this free energy, and also we believe it can be demonstrated by IC50. Of course, it doesn't mean they are good drugs because they could have side effects, other things, but at least they provide a basis for strong binding. We actually provided all these candidates at this website, and also we provide a fragment library. So people can use these candidates to do experimental validation or use these fragments to design their own inhibitors. And for future work, we like to design better approach for predicting chemical synthesis. That is to tell whether it's a chemical reaction is easy or hard. We also like to use the graph neural network to integrate into this work. We actually have done this graph neural network for small molecules to predict properties of small molecules. For example, whether they can be easily insolvent, etc. And we see that graph neural network can be applied here also for better drug design. So that's all I have. Thank you for attention.
Thank you, Dr. Zhu. Thanks for such an informative presentation. We have a couple of questions from our audience. So I'll just go through it and maybe you can answer that. So first question we have is, what do you think about hydrochloroquine that is being recommended by President Trump?
Yeah, of course. Those recommended chemicals. I think they were very promising, but you see that the drug designs have not that easy. Typically, it takes $1 billion to develop FDA-approved drug. So even though there are some promising candidates, I still believe there's room for new development. And in our case, we believe our drug candidates are more potent. So we see that's still meaningful development.
Okay, the other question that we have is, what are some special properties that drugs antivirals need to survive in certain tissues compared with cancer drugs, for instance, solid tumors?
Yeah, that's a very interesting question. So for this virus, I think it's a little bit different from cancer, because cancer typically are localized and they are human cells, and for the virus, first of all, the load could be very big. That means if you look at this COVID-19 viruses, they could be prevalent everywhere. So that's why people found all kind of these symptoms, from losing taste to even neural or psychological effects. So they are very prevalent to a lot of places. That means it is more important to have a potent drug that can really kill those viruses more effectively. And those viruses, here, I think there's a significant difference from cancer. The challenge in cancer is that those cancer cells, they have the so-called somatic mutations that mutate very fast. But this COVID-19 virus, they have a so-called RNA proofreading enzyme. So this molecule, they are highly conserved. I mean, in terms of this RNA sequence or protein sequence. But for some proteins, they have this so-called post-translation modification, like S protein. That's why it's hard to divide. But here, for M protein, the mutations are very few. That means if we find a good candidate to bind the M protein, most likely it can kill the virus effectively, because this virus doesn't really mutate a lot, not like HIV or cancer. So that's why we think if we find one good candidate, it doesn't require like a cocktail approach. Potentially could kill virus very effectively. So that's what I see between the difference between this virus and the cancer.
Thanks a lot for such a detailed explanation. We have a couple more questions. So one is, do anti-SARS-CoV-2 drugs need to work in blood, in lymph nodes, in lung tissues, etc., etc.? Where does it?
Yeah, could you repeat the question again?
Yeah. So do anti-SARS-CoV-2 drugs need to work in blood, in lymph nodes, in lung tissues, etc.? So like exactly where it needs to work on.
Oh, yeah, I think it could be anywhere. So this small molecule, of course, we need to optimize different things. Let's say if you want to target using this approach to target solid tumor or in blood, etc., then you really need to target different things. First of all, it has to be soluble to water. That means it can circulate in blood. And then if you, let's say, target a cancer cell, lymph node, and or other immune cells, then you need to make sure that it actually, if it requires entering the cell, then you need to make sure it can penetrate the cell membrane, things like that. So for the whole drug pipeline, it requires you to consider a lot of things. And also you need to protect it's an enzyme attack. That means that some enzyme can cut the small molecule. So that's why the drug design is very challenging. You need to consider all these factors. But in the drug development pipeline, we are really at the early stage. That means we provide all these candidates. And then you can run additional competition tools to make sure they can be solvent, they can be protected by cleavage of other enzyme, can target certain type of cell, can penetrate the membrane, things like that. So those are the downstream analysis. Yeah.
Okay, thank you. So one more question that we have is, what do you use to dock? What type of docking engine did you use for your pen?
Yeah, so right now we use this commercial system, the Schrodinger. This actually is from it's a company, Schrodinger company. They provide a comprehensive system for docking, for calculating pharmacores, things like that. So down the road, we are thinking to use a more open source tools for docking and others. There are actually plenty of open source docking tools. So by doing that, we can make our system easily usable for academic users. That means they don't have to pay big bucks to Schrodinger in order to use our method.
Okay, so the other question is, how unique is your molecule compared with the molecules in ready to order or ready to synthesize libraries?
Yeah, that's a very good question. So in fact, we actually group our candidates into different categories. The first category is that some drugs are already used, at least without side effects. So that means known drugs. The second category is that we can easily order through a catalog. And third one that we have to synthesize. So we have a combination of all three. And those we need to synthesize, they look quite different from the first two categories. That means they are truly novel. So potentially they could have the open door for a completely different kind of drugs for the same target.
Okay, so I think like there was one question which I might have misunderstood, or the question was about where does the virus live, like the COVID virus on the sick patient? Is it all over the body? Like where exactly it lives?
Oh yeah. So that's actually a very interesting question. So here the common belief that the virus, the S protein, binds to this host protein called ACE2. Although we believe there are additional receptors. In fact, we do separate bioinformatics work to predict those additional receptors. But ACE2 is definitely the primary receptor. And then if you look at the lung cells, they don't even have a lot of ACE2. But those macrophage on lung surface, they have a lot of ACE2. So most likely initial targets for COVID-19 virus is those macrophage. And then through that, they enter into bloodstream, and then they circulate everywhere. So one thing people already see that they attack immune system, like a CD4 T cells and others. So if you look at this lymph node and other immune organs, they shrink dramatically. So that means that they are heavily attacked by the virus. But then once the virus is in the circulation, it goes everywhere. It binds to the neurons. So that's why people found that even this psychological behavior, cognition changed after this COVID-19. And they also have tons of symptoms. I mean, another work we are doing right now is that we are mining Twitter data to categorize all the symptoms reported in the Twitter. Yeah, we found actually many, not only those CDC reported, but pretty much all the organs, for example, some skin, you can have a change of skin color, all kind of symptoms. So that means that they are ubiquitous, they go everywhere. So that's why the load is very heavy. You really require a potent drug to kill the virus.
Okay, thanks, thanks, doctor. Thanks for all your insight about the virus, and thanks everyone for joining in. Since we don't have any question, maybe I'll stop this. And then just wanted to let you guys know that we are now selling the tickets for our 3-day virtual event in June, and the link to purchase the ticket can be found in the reception page of this virtual platform. So please go ahead, and if anyone is interested, please go ahead and check the link. Thanks, Dr. Zhu, again for such an informative session.
Yes, thank you, Mata, and thank you, everyone.